Lipids and lipoproteins. So an introduction, so lipids like triglycerides and cholesterol are not water soluble. That means they can't travel freely in the bloodstream. Instead, the body packages them into lipoproteins, which are spherical particles made of lipids and proteins. There are different types of lipoproteins. So the first one is chylomicrons, which are the dietary transporters. What they carry is they carry triglycerides from the diet. They transport dietary fats from the intestines to the liver, and their role is they're the first step in fat metabolism. These are the largest lipoproteins, but the least dense. So you can think of chylomicrons as delivery trucks that take the fat you eat straight to your liver for processing. Then you have the VLDL, or the very low density lipoproteins. These are the exporters. They carry triglycerides made by the liver. They go to the body cells for energy or storage in fat tissue, and their role is to deliver energy to cells and store the excess. So VLDLs are like suppliers, moving fat made in your liver to where it's needed or where it's going to be stored for later. LDL, which is a low density lipoprotein, the cholesterol carrier, so it carries cholesterol. They go to the cells throughout the body, and its role is to deliver cholesterol used in cell membranes, hormone production, and other cellular functions. LDL is often called the bad cholesterol, not because it's evil, but because too much LDL can lead to plaque buildup in the arteries, which increases the risk of heart disease. And then we have HDL, which is the high density lipoproteins. This is kind of the cleanup crew. So they carry excess cholesterol back to the liver for recycling or removal. Their role is to help maintain balance and reduce cholesterol buildup in the arteries. So therefore, HDL is known as the good cholesterol because it helps protect the heart by cleaning up extra cholesterol. So why cholesterol matters? Cholesterol gets a bad reputation, but it plays vital roles in the body. Structural, it maintains cell membrane integrity. Synthetic, it's a precursor for steroid hormones like estrogen and testosterone, and it acts as a transport, so it aids in the packaging and transport of triglycerides. However, excess cholesterol, especially from LDL, can lead to atherosclerosis, which is plaque in the arteries, which can lead to heart disease and stroke. So coronary heart disease, or CHD, is a condition where the plaque builds up inside the coronary arteries. It reduces blood flow to the heart, and this can lead to chest pain or angina, heart attacks, and heart failure. So how lipids can contribute to congestive heart failure or disease. So high LDL can lead to plaque formation. So low density lipoproteins, or your LDL, deposit cholesterol into your arterial walls. Over time, this forms atherosclerotic plaques, which thickens areas that narrow the arteries. Plaques may rupture, which lead to clots. Therefore, it can cause heart attacks or strokes. Low HDL, so having a low high density lipoprotein, would mean that there's less cholesterol removal. So having a low HDL level means cholesterol is more likely to accumulate. Dietary and fat and atherosclerosis, so high fat consumption, especially saturated fats and trans fats, can increase the risk of elevated LDL, decreased HDL. It can cause inflammation of your arterial walls and atherosclerosis, which again is the hardening and narrowing of your arteries. So atherosclerosis is a risk factor in coronary heart disease. It starts as fatty streaks in your blood vessels and can progress to major blockages and clots. So there are major types of lipids. The body contains several types of lipids, each with their distinct functions. So triglycerides, their function is it's a primary energy source stored in the fat tissue or the adipose tissue. Cholesterol is used in cell membranes, hormone synthesis, and lipid transport. You have phospholipids, which are a main component of cell membranes. And then you have glycolipids, and they're involved in cell recognition and communication. So lipids are an energy powerhouse. Lipids provide more than twice the energy per gram compared to carbohydrates or proteins. Triglycerides are the main form of stored energy in the body. So you can kind of think of triglycerides as your body's batteries, essentially. Structural roles, so you have lipids are essential components of the cell membrane, phospholipids and cholesterol. They're part of the myelin sheath, which is the fatty insulation around your nerves. They also play a role in hormones, so like steroid hormones like your estrogen and testosterone. They are transport of lipids because lipids are hydrophobic. It means they don't like water. They cannot travel freely in the blood. So because of that, they are packaged into lipoproteins, which allow fat and cholesterol to move through the water-based blood system. We've already kind of discussed the other ones as far as chylomicrons, VLDL, LDL, and HDL. Each of those play a different role in the lipid transport. We have lipid chemistry, so triglyceride and phospholipids. Triglycerides, they have a structure of 1 glycerol and 3 fatty acids. Its function is energy storage. It is found in fat tissue, oils, dietary fat. The basic triglyceride structure has the 3 fatty acids, and it's attached to a glycerol backbone. Phospholipids, its structure is 2 fatty acids and 1 phosphate group plus a glycerol. Its function is to make up cell membranes. It is amphipathic, so it has a hydrophilic head and hydrophobic tail, and it forms the phospholipid bilayer of all cells. Triglycerides, so saturated versus unsaturated fatty acids. Fatty acids can be categorized into 2 major types based on their chemical structure and physical properties. Saturated fatty acids contain no double bonds between their carbon atom, which makes them straight and tightly packed. As a result, they are typically solid at room temperature and are mostly found in animal products like butter, cheese, the fatty cuts of meats, etc. On the other hand, unsaturated fatty acids have 1 or more double bonds in their structure. These double bonds create bends or kinks in the chain, and that prevents the tight packaging. That's why unsaturated fats are generally liquid at room temperature. They are predominantly found in plant-based sources like olive oil, sunflower oil, avocados, things like that. Sources of triglycerides. Triglycerides are the main form of fat in the body. They can come from 2 primary sources. The first is an exogenous triglyceride, which are obtained through the diet. So when we eat fatty foods, triglycerides are absorbed in the small intestine and then packaged into chylomicrons, which transport them through the bloodstream to the liver and other tissues. The second source is endogenous triglyceride, which are synthesized by the liver. When we consume more carbohydrates or fat than the body immediately needs, the liver converts the excess into triglycerides. These are then packaged into very low density lipoproteins and sent out into the bloodstream to be stored or used for energy. Triglycerides are not water soluble, so they can't travel freely in the bloodstream. Instead, they are transported by lipoproteins. The chylomicrons, as mentioned earlier, carry dietary or the exogenous triglycerides from the intestine to the liver and other tissues. The VLDL, on the other hand, carries triglycerides that are produced by the liver, so the endogenous, to tissues around the body. Both of these lipoproteins serve to deliver triglycerides to tissues where they can be either stored in adipose or fat tissue or used for energy in muscle cells. The breakdown of triglycerides or lipolysis. Once triglycerides reach target tissues, they need to be broken down into usable components. This process is called lipolysis. The main enzymes involved include lipase, a general enzyme that breaks down fats in various tissues, and lipoprotein lipase, which is found on the walls of blood vessels and acts specifically on triglycerides within lipoproteins like chylomicrons and VLDL. LPL breaks triglycerides down into glycerol and free fatty acids, which can then be taken up by the cells. Hormones also play a major role in triglyceride breakdown. So epinephrine, also known as adrenaline, is released during stress or exercise and stimulates fat breakdown to provide quick energy. Cortisol is a hormone associated with long-term stress and fasting. It also promotes the breakdown of fat stores to ensure energy availability when glucose is limited. So the flow of fat metabolism, the process, here's how it works basically. When you eat fat, it's broken down into triglycerides, which are then packaged into chylomicrons and released into the bloodstream. Lipoprotein lipase then breaks the triglycerides down into fatty acids, which are absorbed by body cells and either burned for energy or stored as fat. When the liver makes triglycerides from excess nutrients, it sends them out in the form of VLDL. Just like with dietary fat, These VLDL particles are acted on by lipoprotein lipase, which helps free the fatty acids for use or for storage. So cholesterol, we've kind of already talked about it, but let's go a little bit deeper. So what is it? Cholesterol is classified as an unsaturated steroid alcohol. It's a lipid, but unlike triglycerides and phospholipids, it has a ring structure rather than long fatty acid chains. This structure makes it uniquely suited for both structural and biochemical roles in the body. So cholesterol, it is found in all cells and body fluids. It is a vital component of cell membranes where it helps regulate fluidity and stability. Without cholesterol, our cells would be too rigid or too permeable, both of which can disrupt cellular function. In addition to structural roles, cholesterol is crucial for several biosynthetic pathways. So it serves as a precursor for bile acids, which help digest fats in the small intestine, vitamin D, which is important for calcium absorption and bone health, steroid hormones, including sex hormones like estrogen and testosterone, as well as the adrenal hormones like cortisol and aldosterone. Cholesterol with the diet. So humans consume about 100 to 700 milligrams of cholesterol per day from the diet, depending on, your eating habits. Foods high in cholesterol include egg yolks, organ meat, shellfish, and full-fat dairy. However, the body doesn't rely solely on dietary cholesterol. Actually, the liver synthesizes between 500 to 1,000 milligrams per day, often more than what we consume. So this internal production ensures that cells have a steady supply of cholesterol even when dietary intake is low. This also explains why dietary cholesterol has less of an impact on blood cholesterol levels than once believed. So the body can adjust its own synthesis in response to dietary intake. So lipoproteins, these are molecules that are responsible for transporting fats like triglycerides and cholesterol through the bloodstream, and they can be classified in two main ways. One by density, and this refers to the ratio of lipid to protein content. The more protein a lipoprotein has, the higher its density, or by the electrophoretic mobility. This technique separates lipoproteins based on how they move in an electric field, which depends on their size and charge. So both methods characterize lipoproteins into four major types. So we got the chylomicrons, the VLDL, the LDL, and the HDL. Each type plays a different role in lipid transport and metabolism. So once again, chylomicrons are the largest of all lipoprotein particles. Their large size and extremely high fat content make them easy to distinguish. They are the primary carriers of exogenous triglycerides. So that's the fat, if you remember, that was absorbed from the diet through the small intestine. Chylomicrons are composed of 90 to 95% triglycerides, with the remainder made up of cholesterol, phospholipids, and proteins. After a fat-containing meal, chylomicrons enter the lymphatic system and then the bloodstream, where they deliver dietary triglycerides to various tissues for storage or for energy use for later. Because chylomicrons are only present after food intake, they are normally not found in a fasting blood sample taken after 12 to 14 hours without eating. In fact, their presence as a fasting sample can indicate a problem with fat metabolism. One easy way to recognize chylomicrons in a blood sample is the formation of a creamy layer at the top of the serum or plasma after an overnight refrigeration period. This creamy layer floats due to the extremely low density and high fat content of chylomicrons. So VLDL is a very low density lipoprotein. It's one of the primary types of lipoproteins in the body. So unlike chylomicrons, which carry dietary or the exogenous fat, VLDLs are the major carriers of endogenous triglycerides, meaning these are the triglycerides that are produced by the liver. The VLDL particles are composed of about 50 to 65% triglycerides, along with cholesterol, phospholipids, and proteins. So because of their high triglyceride content, they are relatively low in density compared to LDL and HDL. So with the diet in VLDL production, so although a VLDL carries fat synthesized by the liver, dietary habits do have a direct impact on how much VLDL is produced. When a person consumes excess calories, especially from refined carbohydrates and fats, the liver increases triglyceride synthesis. This rise in liver fat production leads to an increase in VLDL production. So in other words, even though VLDL transports endogenous triglycerides, its production is highly influenced by exogenous dietary intake. So more dietary intake means more liver fat production, which means more VLDL, which is going to be released into your bloodstream. So an increase in VLDL is often detected through a rise in blood triglyceride levels. Since VLDL is rich in triglycerides, elevated VLDL production contributes directly to hypertriglyceridemia, a condition where triglyceride levels in the blood are abnormally high. High levels of VLDL are considered a risk factor for atherosclerosis and cardiovascular disease, especially when accompanied by a low HDL and a high LDL level. LDL, it's a low-density lipoprotein. It's one of the most well-known lipoproteins because of its strong association with cardiovascular disease. LDL is responsible for transporting cholesterol from the liver to peripheral tissues throughout the body. This delivery function is important because cells need cholesterol to maintain membranes and produce hormones. LDL particles are approximately 50% cholesterol by weight, making them cholesterol-rich carriers in the bloodstream. The rest of the particle is made up of phospholipids, proteins, and a small amount of triglycerides. So why is LDL considered the bad cholesterol? Although cholesterol is essential for many biological functions, excess LDL in the bloodstream is harmful. High levels of LDL lead to cholesterol buildup in the walls of the arteries, contributing to the development of atherosclerotic plaques. Over time, this can narrow the arteries, reduce blood flow, and increase the risk of coronary heart disease, heart attacks, and strokes. So because of this strong link between elevated LDL levels and cardiovascular risk, LDL is often referred to as the bad cholesterol. HDL is high-density lipoprotein, and this is often referred to as the good cholesterol, and really for a good reason. The HDL particles contain a high proportion of apolipoproteins, making up of about 45 to 60% of their total weight. These proteins not only give HDL structural support, but also play an active role in lipid transport and metabolism. So the primary function of HDL is to collect excess cholesterol from tissues and the bloodstream and transport it back to the liver. This process is known as reverse cholesterol transport. And once in the liver, the cholesterol can be reused, converted into bile acids, or excreted from the body. By removing the cholesterol from peripheral tissues and preventing its buildup in blood vessel walls, HDL helps protect against atherosclerosis and cardiovascular disease. So because of its role in that cleaning up the excess cholesterol, HDL helps reduce the risk of the plaque formation and supports overall cardiovascular health. Usually higher HDL levels are associated with a lower risk of coronary heart disease, and because of that, HDL is labeled the good cholesterol. It helps keep the blood vessels clear and the heart healthy. Lipid synthesis, so triglyceride synthesis. Triglycerides are the primary form in which fatty acids are stored inside cells. To synthesize triglycerides, fatty acids first undergo activation, where they are converted to acyl-CoA esters, a necessary step for their incorporation into triglyceride molecules. This activation process enables the fatty acids to combine with glycerol backbones to form triglycerides, which are then stored as energy reserves within the adipose or fat tissue or used for cellular functions. Cholesterol synthesis, on the other hand, the liver is the major site of cholesterol synthesis in the body. Cholesterol produced by the liver is essential for various physiological functions, as we mentioned before, including maintaining cell membrane integrity, producing bile acids, synthesizing vitamin D, and serving as a precursor for those steroid hormones. The liver tightly regulates cholesterol synthesis to balance dietary intake and bodily needs, ensuring adequate supply without excess accumulation. Lipid metabolism, so the exogenous lipid pathway refers to the transport of dietary lipids absorbed from the small intestine. After a meal, dietary fats are packaged into chylomicrons, which can carry triglycerides through the lymphatic system and bloodstream back to the liver. This process ensures that dietary lipids are delivered for energy use, storage, or for further metabolism. The endogenous pathway, on the other hand, manages lipids produced by the liver. The liver synthesizes triglycerides and cholesterols, which are then transported through the bloodstream by VLDL and LDL particles to peripheral tissues. This system supplies fats and cholesterol needed for cell membranes, hormone synthesis, and energy storage. The reverse cholesterol transport pathway utilizes HDL to collect excess cholesterol from peripheral tissues and carry it back to the liver. Once returned to the liver, the cholesterol can be reused, converted into bile acids, or eliminated from the body. This pathway is critical in maintaining cholesterol balance and protecting against cardiovascular disease. Effect of hormones. So we have insulin. Insulin plays a key role in regulating lipid metabolism, especially in the liver. It regulates the synthesis of VLDL, the lipoprotein that transport the endogenous triglycerides. When insulin levels are high, it can lead to increased plasma triglyceride levels and a decrease in HDL cholesterol. Additionally, insulin influences cholesterol metabolism, often resulting in changes in blood cholesterol levels. Growth hormone can stimulate lipid metabolism by increasing the production of VLDL by the liver. This means more triglycerides are transported to the peripheral tissues, supporting energy needs during periods of growth or stress. Thyroid hormones also affect lipid profiles, particularly by influencing LDL levels. Elevated thyroid hormone levels are associated with an increase in LDL cholesterol, which can impact cardiovascular risk depending on the overall lipid balance. To measure cholesterol, so the cholesterol measurement, the reference range for cholesterol measurement is the Lieberman-Burkhard reaction. It's a classical chemical test. In most labs now, we're going to use enzymatic methods, which are more commonly used. These involve enzymes that first cleave fatty acids from cholesterol esters, which converts them into free cholesterol. The free cholesterol is then acted upon by cholesterol oxidase, which produces hydrogen peroxide. This hydrogen peroxide reacts further to form a colored compound, and then this color, the intensity of it, is how we measure it by a spectrophotometer to quantify the cholesterol levels. Triglycerides are measured in enzymatic reactions as well. Initially, lipase cleaves the fatty acids from the glycerol backbone. The free glycerol is then processed by enzymes such as glycerol kinase and pyruvate kinase. This series of reactions leads to the conversion of NADH to NAD+, which can be measured using UV spectrophotometry to determine triglyceride concentration in the blood sample. Lipoproteins can be separated and quantitated by different methods, and some have pros and cons, right? So ultracentrifugation, this technique separates lipoproteins based on their density by spinning samples at very high speeds. The different lipoprotein fractions form layers, allowing their collection and measurement. Next one is electrophoresis. So lipoproteins can be separated by their electrophoretic mobility in an electric field, where they migrate at different rates based on size and charge. Chemical precipitation. This method uses chemicals to selectively precipitate certain lipoproteins from the serum, making it easier to measure the remaining fractions. So for HDL measurement, chemical precipitation is commonly employed, followed by cholesterol analysis on the supernatant. However, it's important to note that increased triglyceride levels can interfere with the sedimentation of precipitates, potentially affecting the accuracy. So calculating LDL and VLDL, since LDL is not always directly measured, it is often calculated using the formula shown below. It's the LDL cholesterol equals the total cholesterol minus the HDL minus the VLDL. The VLDL calculation is just triglycerides divided by 5. So this formula is widely used in clinical settings, but is most accurate when the triglyceride levels are within a normal range. Quality control, as always. So the quality control material consists of pools of freshly collected patient serum. These serum pools are aliquoted into sealed vials, quickly frozen and stored at -70 degrees Celsius to help preserve their integrity. This approach ensures consistent and reliable control samples for lipid assays over time. So just like with every other thing else we do in the lab, there's quality control involved for these tests as well. Population levels can be different for cholesterol. So women generally have higher HDL levels and lower triglyceride and cholesterol levels compared to men. Both men and women experience an increase in cholesterol, LDL, and triglycerides as they age. Young children have lower lipid levels than adults, and men tend to have lower HDL and higher LDL cholesterol and triglycerides, making them more prone to heart disease. So some disease information. So arteriosclerosis is one of the leading causes of death and disability worldwide. So trying to understand its pathology, effects, and associated diseases is obviously crucial for diagnosis and treatment. So the pathophysiology for arteriosclerosis is characterized by the formation of plaque within the arterial walls. These plaques originate from fatty streaks, which are accumulations of lipid-laden macrophages. Over time, plaque buildup narrows the blood vessels, reducing blood flow and leading to ischemia. This narrowing can impair oxygen and nutrient delivery to tissues, contributing to various clinical conditions. So, those conditions. Peripheral vascular disease affects the arteries in the legs and arms. The narrowing of these arteries reduces blood flow to the extremities, causing pain, numbness, and sometimes tissue damage. Risk factors for this would include smoking, diabetes, and high cholesterol. Another disease would be coronary artery disease. This involves the coronary arteries supplying blood to the heart muscle. The plaque buildup can cause angina, which is chest pain due to reduced blood flow. Severe obstruction may lead to myocardial infarction or a heart attack, resulting from prolonged ischemia and heart muscle damage. The next one is cerebrovascular disease that can affect the arteries supplying the brain. Narrowing or blockage can cause strokes, leading to neurological deficits depending on the area of the brain affected. Prevention includes managing risk factors such as hypertension, smoking, and hyperlipidemia. Deposits in the vessel walls are strongly associated with increased levels of low-density lipoprotein cholesterol and decreased levels of high-density lipoprotein cholesterol. LDL cholesterol is often referred to, as we mentioned, as the bad cholesterol because it contributes to the plaque buildup, while HDL is considered the good cholesterol because it helps remove cholesterol from the bloodstream. So I remember this by LDL. It's the bad. We want that to be low, so we got L for LDL. And then HDL for the good. We want that to be high for HDL. It's just one of those. That's how I remember it. So if it helps you, there you go. Diagnostic evaluation. It is essential to measure total cholesterol, HDL, and LDL levels in patients. These measurements help to assess cardiovascular risk and determine whether lifestyle changes such as diet modifications or pharmacological interventions are needed. Proper lipid profiling guides clinicians in managing patients at risk of arteriosclerosis and related diseases. Some lipid disorders and their implications. So we have hypercholesterolemia. It is defined as high levels of cholesterol in the blood. It is closely linked to the development of heart disease due to the promotion of plaque formation in the arteries. Management often involves dietary changes, exercise, and cholesterol-lowering medications like statins. Hypertriglyceridemia is characterized by elevated triglyceride levels. It is life-threatening in severe cases because it can lead to pancreatitis, which is an inflammation of the pancreas. Causes include a deficiency of the lipoprotein lipase, which is that enzyme that is essential for breaking down triglycerides, and also a deficiency of the apolipoprotein C2, which is a cofactor necessary for the LPL function. And then we have hypoalpha-lipoproteinemia, my goodness, bear with me here. It's characterized by a decrease in HDL cholesterol levels. Since HDL is responsible for removing cholesterol from the bloodstream, its reduction is linked to an increased risk of premature coronary heart disease. So patients with low HDL levels have a higher likelihood of developing atherosclerosis due to impaired cholesterol clearance. And then we have hypo-beta-lipoproteinemia, which is marked by a decrease in LDL cholesterol levels. While low LDL is generally considered protective against heart disease, this condition can sometimes be associated with other metabolic issues depending on the underlying cause. It is less common, and it does require careful clinical evaluation to determine that that's what it is. So both of these conditions highlight the importance of balanced lipoprotein levels for cardiovascular health, and being able that we need to monitor the HDL and LDL concentration that's able to help identify individuals that are at risk, maybe ones that we need to get them on some preventative, like exercise and or therapeutic interventions as well.